Okay, this is a screencast to basically get us caught up. We're a little behind because of academic convocations and things like that. So I want to give you an introduction to translation that will get us kind of on track. So in this chapter, the things that you will need to learn and be held responsible for is knowing the process by which messenger RNA is used to create amino acid sequences. We've talked about this in general terms before. We'll talk about it in more specific terms now. You'll need to be able to describe the structure of the ribosome, transfer RNA, and how those structures relate to the function of those molecules. You'll need to explain the couple of examples of translational regulation in eukaryotes that are described in your textbook. There's also a section of your textbook that deals with redundancy in the genetic code, but we've already talked about that in some detail up to this point. I'll touch on it again, but not in this screencast. We'll cover that in class. And then there is processing of proteins after the initial amino acid sequence is laid out that are important in proteins achieving their final three-dimensional form so that they're functional. And these are post-translational processing mechanisms, and we'll also talk about those as well. But once again, not in this screencast. In this screencast, we're basically going to concentrate on the first three of these things. So going back to the central dogma, once again, the central dogma involves the transcription of messenger RNA, and I prefer that you put a little M in front of RNA because DNA is transcribed into messenger RNA, and that messenger RNA is then translated into a polypeptide. It doesn't mean that DNA doesn't get transcribed into other forms of RNA, but in terms of this route from DNA to messenger RNA to polypeptide, this is more precisely messenger RNA, not ribosomal RNA, not transfer RNA, not small interfering RNAs, etc. When you transcribe the messenger RNA then specifies a sequence that then is translated into the amino acid sequence in a polypeptide by transfer RNA. This polypeptide then goes through post-translational processing to get folded up into the final functional form of the protein, and that is what essentially, hopefully, affects phenotype. And so when we think about translation, messenger RNAs are going to be the template by which the amino acid sequence is determined. In bacteria, this is all done at the same time as transcription. Transcription makes the messenger RNA, and as soon as that messenger RNA starts getting made, the messenger RNA starts being translated. That can be done because there is no division between where the genetic material is and where the ribosomes are for making proteins. So when you don't have a nuclear membrane surrounding your nuclear material, the bridge between transcription and translation is seamless. In eukaryotes, though, remember that transcription occurs in the nucleus and translation occurs in the cytoplasm. And so you have to transport the transcript, the messenger RNA transcript, out of the nucleus into the cytoplasm. And remember that this is facilitated by the addition of a 7' methylguanine on the 5' end of a messenger RNA molecule. That serves as a signal to the nuclear membrane to allow transport out of the nucleus into the cytoplasm. Once it's out in the cytoplasm, the ribosome will bind to the 5' end of the messenger RNA, and transcription will proceed. So when you think about translation in a general sense, you need to know that translation proceeds from the 5' end. Here you see the start codon to the 3' end of messenger RNA, but the order of amino acids is determined ultimately by these 3-letter codes called the codon. And we've talked about the codon code in the past, and each of these codons specifies a specific amino acid. AUG, adenine uracil guanine, always specifies for methionine. And these amino acids are essentially brought to the messenger RNA molecule by these transport RNAs called transfer RNAs, tRNA. And each of these tRNAs has a different structure, and that structure allows it to bind to a different type of protein. So however many, a different type of amino acid. So however many amino acids you have, you're going to have that many different kinds of transfer RNA molecules. They will each have a different codon, and that codon complementary base pairs with the, has an anticodon rather, that anticodon complementary base pairs with the codon of messenger RNA, and that is what specifies the sequence of amino acids. When you have the first codon in here, it's already had methionine added to it. There's a second amino acid here. Then another transfer RNA molecule will complementary base pair with the next codon using the anticodon. Then that amino acid is bound to the second amino acid in this now 3 amino acid chain. And this 3 amino acid chain then dissociates the second amino acid from that first transfer RNA molecule that is there, and then that thing gets kicked out, and this process of elongation continues. So when we look at the structure of an amino acid, there are a long sequence of RNA nucleotides. In some cases, they complementary base pair with one another, and in other cases, they're free. One of the free areas is this anticodon down here at the bottom, and then there are also free nucleotides, free nitrogenous bases exposed up here as well. So you have areas of nitrogenous bases exposed here, here, and here. The ones that are important are the anticodons and the ones up here where the amino acid attaches. The amino acid is attached via aminoacyl synthetase, which basically takes the amino acid and binds it to the free end of the transfer RNA molecule. So this transfer RNA molecule fits inside of 3 different active sites in the ribosome. There's a peptidal site, there's an aminoacyl site, and there's an exit site. One codon is exposed in each of these sites, and so this is where all of the activity of transfer RNA interacting with messenger RNA occurs. Ribosomes, though, have their own structure, and these ribosomes are made up of ribosomal RNA. The sizes of the ribosome, it comes in a large subunit and a small subunit, and these subunits actually exist separately until they converge on a messenger RNA molecule, and they essentially clamp a messenger RNA molecule in between these 2 subunits. The subunits are different sizes in bacteria and eukaryotes, but they're basically made up of a bunch of constituent RNA molecules and associated proteins. These RNA molecules, ribosomal RNA molecules, you will remember from past chapters, are basically created using RNA polymerase I rather than RNA polymerase II. Remember that RNA polymerase II is what is responsible for creating messenger RNA. RNA polymerase I is what is used for creating ribosomal RNA subunits. These subunits interact with proteins to basically form the 2 large subunit and small subunit. When you look at transfer RNA, you can envision it any way you want. You can make these ribbon molecules. You can make other diagrammatic ways of thinking about this. The important thing, I think, to know about transfer RNA is that you have free nucleotides, free nitrogenous bases at one end. You have free nitrogenous bases down here. These nitrogenous bases form the anticodon that basically allow complementary base pairing with the codon, and at this 3' end up here, that's where the amino acid is connected. Also remember that each transfer RNA molecule is specific to a particular amino acid. So the transfer RNA molecule that bonds to lysine is going to be different in structure from the transfer RNA molecule that bonds to isoleucine. And so you have to have as many transfer RNA molecules as you have amino acid substrates that they're going to be directing. And so this is how your book often, this one on the far right is how your book uh, essentially draws these things, because it has kind of the basic structure and it concentrates on the anticodon sequence, which is important for complementary base pairing and getting the messenger RNA to tell the ribosome what order to put these amino acids in. So we've talked about the redundancy in the code. Remember that there's only one start codon and there are 3 stop codons. The stop codons are stop codons because basically the codon codes for a transfer RNA molecule that is never produced by the cell. And so it isn't that there's a transfer RNA molecule that comes in and stops replication. It's just that when you get to a uracil adenine adenine, a uracil adenine guanine, or a uracil guanine adenine, there is no transfer RNA molecule produced that has an anticodon complementary to the stop codon. So there is no transfer RNA molecule. There is no peptide bond built between the previous amino acid and the new amino acid, because there is no amino acid there, and that's what signals stopping of translation. And so when you look at the anticodon, you can't find any transfer RNA molecules that have stop codons associated with them. So when we look at elongation, and we're going to put off initiation for a few minutes, but when you look at elongation, what happens is a molecule comes into the aminoacyl part of the ribosome. There's already a transfer RNA molecule occupying the peptidyl site, and there is a transfer RNA molecule that is here in the exit site of the ribosome, but it is being kicked out because it has already lost its serine and is uncharged, and it exits the ribosome and then goes off and gets charged with aminoacyl synthetase. This act of adding an amino acid to a transfer RNA molecule is referred to as charging the transfer RNA molecule, and so one that has lost its amino acid because it's gone through this process basically is referred to as an uncharged molecule, and the enzyme that charges the transfer RNA molecule is called aminoacyl synthetase. And so in this figure here, this one is uncharged. The uncharged one is being released from the ribosome. The transfer RNA in the peptidyl site in the ribosome has the previous amino acid attached to the amino acid that is attached to the transfer RNA, and then the newly arrived transfer RNA containing threonine is then going to be bound to the aspartic acid amino acid here. At that point, there will be a peptide bond linking threonine and aspartic acid and serine. At that point, the bond between the transfer RNA molecule will be broken. These things will be attached to this transfer RNA molecule. The ribosome as a whole will shift to the right, or that you can think of the messenger RNA as shifting to the left. This aspartic acid transfer RNA molecule will then move to the exit site. That will move the threonine containing transfer RNA molecule to the peptidyl site. The aminoacyl site will be open, and that will allow the arrival of a new amino acid over here containing isoleucine, carrying isoleucine, and you'll go through the process again, forming a peptide bond between the two amino acids, breaking the bond with the transfer RNA. The whole thing shifts down, moving the isoleucine to the peptidyl site, moving the threonine. That is threonine. They misspelled it in the book. Moving the threonine transfer RNA to the exit site, it gets kicked out, and then a new molecule will come in. This is the process of elongation. It's not that complicated. How it all gets started is basically at the beginning of translation. These two molecules, the large subunit and the small subunit, are existing unseparated. And when the presence of messenger RNA with a start codon and a 7' methylguanine cap are there, basically recruits the two subunits to essentially clamp around the messenger RNA molecule. So the messenger RNA molecule is actually going through this tunnel through the middle of these two subunits. And there is a special methionine transfer RNA molecule that will come in, and while most transfer RNA molecules will enter into the ribosome at the aminoacyl site, methionine, these initiation transfer RNA molecules, which are slightly different than regular methionine transfer RNA molecules, actually comes in and resides in the peptidal site, links to the start codon, and once that happens, that then allows a second codon to come in, occupy the aminoacyl group and the aminoacyl site, and then you form the peptide bond, break the bond of methionine with its transfer RNA molecule. The whole thing shifts down, methionine gets kicked out, and that's how it all starts. But it all begins at this start codon in the peptidal site of the ribosome of the ribosome subunits. And so there are proteins that facilitate this initiation, and but it's much more straightforward than, for example, transcription. There can be translational regulation, which we'll talk about in a little while, where you do have molecules adding on to the end that can either prevent the beginning of translation, or can serve to stabilize or degrade the messenger RNA molecule, as we talked about with things like interfering RNAs and things like that. But this is how it begins. It begins by the recruiting of both subunits of the ribosome, the recruiting of methionine into the peptidal site. Once that occurs, then the process of elongation begins. Once again, translation gets terminated because the stop codons code for a set of anticodons that don't exist in transfer RNAs. So we don't create transfer RNAs that are complementary to this, and so no new transfer RNA comes in here and complementary base pairs, so there's no amino acids to connect to that, and so that basically puts a stop to translation. Now, there can be mutations that occur in the DNA that codes for transfer RNA, and in some cases, those mutations allow a transfer RNA molecule to have an anticodon that matches a stop codon, in which case you don't have the stopping of translation where it should, and that ends up creating a protein that is in many cases non-functional because it produces a much longer protein, and everything after that doesn't actually have any meaning, in the sense that it doesn't define a functional protein. It's just this untranslated region that occurs after the stop codon that gets translated when it shouldn't be translated, and that generally affects the structure and function of the resulting polypeptide when it goes to fold up. It doesn't fold up correctly. When it doesn't fold up correctly, it doesn't carry out its function. So your book talks about translational regulation. Not a lot is known about translational regulation, but essentially there are a couple of well-studied examples, and your book talks about both of them. The first one of them has to do with iron presence in the body. One of the problems with iron, while we need iron for things like hemoglobin, if we have too much iron in our bodies, it becomes toxic, and so we have to have ways of regulating iron. And the proteins that basically help with this are of two types. One protein is called ferritin. Ferritin is basically a protein that binds to iron, and in the process of binding to iron, basically removes it from freely circulating in our bloodstream. It makes it not absorbable any longer. And so the other one of these is called transferrin, and transferrin is a protein that allows the cell to import iron into the cell. And so these both act as kind of antagonists to one another. So in the case of ferritin, what happens is when iron is high in the body and you need to basically reduce its toxic impact, you need to somehow tie it up, bind it up so that it doesn't interact with things. And so ferritin is actually going to interact with iron to essentially protect the body from absorbing it. What happens in this method of regulation is you have an iron response element here at the beginning of the messenger RNA. This is in the 5' untranslated region of messenger RNA. Remember that when you transcribe RNA, there's an untranslated region at the 5' end, there's an untranslated region at the 3' end, and this is a regulatory part of this messenger RNA molecule. So there's what's called an iron response element. And what can happen is there can be an iron response protein that binds to the iron response element. That binding of that to the binding of the IRP to IRE essentially shuts down translation of this gene. Basically interferes with translation. Translation is blocked, no ferritin is produced. This is a situation in low iron environments. So anytime your body has a normal amount of iron, the iron response protein is bonded to the iron response element. That shuts down translation, no ferritin is produced. When iron is in great abundance in the body, iron interacts with this iron response protein. That interaction basically removes that from the iron response element. Once that is removed, the messenger RNA is freed up to do translation. Translation occurs, ferritin is produced, and this excess iron gets basically bound to ferritin and is no longer available for absorption, and you essentially reduce the levels of available iron in the bloodstream. Transferrin operates in a different way. Transferrin, once again, is a protein that allows transport of iron into the cell. And so this is going to actually want to be made when you need more iron. So this is going to be responding to low iron conditions. And transferrin basically involves, once again, a bunch of iron response elements, 3 iron response elements. Only these are located in the 3' untranslated region of the messenger RNA. And what happens is that when these structures are here, when IRE has iron response proteins attached to them, it basically serves to stabilize the messenger RNA molecule. When the messenger RNA molecule is stabilized, it tends to not degrade as fast. Remember, we talked about how messenger RNA is a transient molecule. It tends to be made very quickly, and it tends to be degraded very quickly. And so these iron response elements, when the IRPs, the iron response proteins, are bonded to them, stabilizes messenger RNA so that when it does get made, it's retained in the cell for a longer period of time. This then allows this messenger RNA molecule to continue making transferrin. This transferrin then allows the transport of iron into the cell when cells need iron. However, when iron is in high abundance, once again, these IRPs, these iron response proteins, basically complex with iron, that removes them from these iron response elements. The stabilization of messenger RNA is removed, and as a result of that, the messenger RNA breaks down, and so you get less translation of transferrin. And so you have less transferrin, you have less iron being brought into the cells, because iron in too high a level is toxic to cells. So these are just 2 examples of how protein abundance can be affected not by transcription of messenger RNA, but by translation of messenger RNA into the final protein. And with that, we'll end the screencast, and we'll talk about everything else that is in this chapter in class on Wednesday.